Hello everyone, my name is Zheng Ning. I'm from Karolinska Institute. The topic of my presentation today is high-definition likelihood inference of genetic correlations across human complex traits. So this study has been published on Nature Genetics, and we made an R package called HDL to implement the method we developed in this paper. So traditionally, when we have individual level genotype data, we can use GREML to estimate genetic correlations. However, GREML can be computationally intensive, especially when the number of sample size is large. That's why LD score regression, or LDSC, became very popular. So basically, LDSC developed the relationship between the variance of test statistic and its LD score. So when we have two phenotypes, we can extend this equation to estimate genetic covariance and the genetic correlation. Although LDSC and GREML are both based on the same linear mixed model, people have realized that LDSC estimates is much less precise than GREML estimates. So we were thinking there must be something missing in LDSC. Then after some analysis, we realized that not only the variance of the test statistic is determined by the LD matrix, actually the whole variance covariance matrix of the test statistic is determined by the LD matrix. So if we determine R, if we denote R as the LD matrix, and we define LD score matrix L as the self-production of R matrix, then we can derive the relationship between the variance covariance matrix of the test statistic and the L matrix. Interestingly, if we look into the L matrix, then the diagonal elements are exactly the LD scores, which means that LDSC only uses the diagonal elements, the diagonal information in the L matrix, and it ignores all of the off-diagonal information. So we were thinking if we are able to use all of the information, we should be able to get more precise results. Similar to LDSC, when we have two phenotypes, we can estimate genetic correlation. So we apply both of these two methods. Firstly, in simulation, we simulated quite a lot of different scenarios, and in all of the scenarios, HDL is more efficient than LDSC, or the standard error from HDL estimates is less than LDSC estimates. And in all of the scenario, HDL is at least 60% more efficient than LDSC, which is equivalent to 2.5-fold increase in sample size. And then we apply these two methods to UK Biobank to estimate the genetic correlation between 30 phenotypes. And then this is the result. The upper triangular shows the LDSC results, and the lower triangular shows the HDL results. And those genetic correlations, which are significant in both methods, are marked with a dot, and those ones which is significant in only one method is marked with an asterisk and a square, black square. We can see that there are much more significant results in lower triangular, which is HDL result, because HDL estimates has lower standard errors. So in summary, HDL only requires G1 summary statistics to estimate genetic correlation, but it's more precise than LDSC. And it takes about 10 to 15 minutes to estimate one genetic correlation, but it can be easily parallelized by using our R package HDL, which is hosted on GitHub. And if you are interested in more details about HDL, you can read our paper. Thank you.